And then it's my great pleasure to introduce you to the speaker of this lecture today, Professor Javier Montenegro from University of Santiago de Compostela, who is going to tell us today about supramolecular chemistry and all the beautiful work I think that they have done in this field. But I think starting with a bit of a background in what is supramolecular chemistry and how can we use it in synthetic cell research. Very briefly, Javier did his PhD also in Santiago de Compostela, but after that he spent time in many labs, including in Cambridge with Steve Lay, at Scripps with Reza Kadiri, and later also in Geneva with Stefan Mathil. And then I think in 2009 he came back to Santiago de Compostela to start his group, and has been very successful in doing all kinds of fantastic work with supramolecular chemistry, mostly peptide fibrils since then. And I look forward to the presentation. Javier, please, the floor is yours.
Thanks so much, Evan, for this very kind introduction and for the invitation to this series of lectures, which are very interesting for our field, for our common shared interest. And of course, thank you very much for taking care and organizing all this, because we know, all of us, how much time it takes. And so we are all very thankful for you for taking care of organizing this lecture series. So I have rephrased a little bit the title of the talk. It's one of the first times I'll be only speaking about this part of research in our group. And I have, because although we are in a sort of common shared interest in synthetic cells, I think many or most of the people who's interested in replicating a living cell is also interested have interest in the origin of life kind of questions, and why some things were and how some functional behaviors emerge at the very beginning. And so I have wrote a new kind of simplified title for the talk, which will be I will be speaking about how peptide amphiphiles can be mimicking a role of functional protofilaments that have maybe play a role at the beginning of the beginning of life. And so recently, I like to start my talks by showing this molecule and by asking one question to the attendees. I don't know how it will work online, but I'm doing it all the time, so I will do it now as well online. So this is a double-stranded DNA in a V form, the classical structure of this molecule. And I'd like to ask you, what do you think is the non-covalent interaction that it's in terms of energetic contribution weighing more to stabilize DNA into this particular structure? So sometimes, if you want to ask, you can unmute yourself and maybe participate, although in presence is easier. But I think that in many occasions we can think about the hydrogen bonds between the nucleobases, because this is what we study and this is what we know. They are directional, they are something that we can understand well, the number of hydrogen bonds, the stability, and so on and so forth. But in fact, the most important energetic contribution is due to the hydrophobic effect. And this has been measured in different papers. For example, in this one here, what they have done is they use optical tweezers to dehybridize the DNA and check the stability in the presence of different compounds and solutions. But in this very old paper, they found a way to determine that this hydrophobic effect importance very simple. What they did was to measure the melting temperature of the double strand in the presence of a small addition of co-solvents. So they found that addition of hydrogen, small amounts of hydrogen disrupting solvents such as DMSO or DMF have very little to no effect on the stability of the double strand. However, if they were adding solvents that could affect the hydrophobic the hydrophobic effect, like in the series methanol, ethanol, and isopropanol, this was a really dramatic effect on the stability and was affecting a lot the formation of this particular structure. And so if we think on the thermodynamics of this assembly process, you might know that this hydrophobic effect emerges from the orientation of the water dipole moments nearby the structure of a hydrophobic molecule. And when these two hydrophobic molecules they come together, they will expel these two solvent molecules out of the hydrophobic cavity. And if this happens with many, many molecules of water, then you have a strong stabilization. So this is important because it means that the hydrophobic effect is entropically driven, right? So it has some important consequences. So in first place, the hydrophobic effect is solvophobic, so it's not something directional. It's more depending on the solvent that you have. And on the other hand, it's very difficult to quantify and to measure the hydrophobic effect and the hydrophobic interactions. So whereas some other non-covalent forces are more easily detectable and quantified, the hydrophobic effect is complicated to quantify and to put numbers on it. And this has consequences, but it is, I think, for everybody of us working in synthetic cells or at the origin of life kind of research programs, we know that the hydrophobic effect was very important, critical at the very beginning of life. And this has been work in the work of many others, such as Pierluigi Luisi or Jacques Sostak, how the formation of amphiphiles was key to, or could have been key for the assembly of the first membranes that would allow the formation of first compartments where more complex processes could have started. And well, I will accept that thing. I use this slide to exemplify the important role of the bottom-up approaches towards the replication of cells or the replication of functions at the very beginning of life. Well, first of all, I would recommend everybody in this audience to read this paper here. This is not a research paper, it's a perspective or a viewpoint paper at American scientists, and this is a very nice paper in where they basically talk about the importance of function as the cause of life. And I think that maybe some of us, or maybe much of us share this some sort of, I would say, deterministic visions as chemists of the potential origin of life, because we go to the lab and we mix stuff and we hope molecule A will react with molecule B to exert a function to do something. And so for us, this is also very, very motivating, this kind of vision that the function is what determines the emergence of lifelike or living systems. And so I like to use this slide to show the importance of starting from the real bottom, right? Try to reconstruct living or lifelike systems with using the simplest building blocks that we can access, and actually, because as chemists we can make them to understand how modifications could affect them as well. And so, for example, here I use this to exemplify that if you want to understand the transportation system of a big city, looking from the top to the freeway interconnected image like this one, it's going to be very, very difficult for you to try to understand all these processes, all these connections, all the moving parts. It's going to be kind of a mess. But if you minimize this to the functional question, why these guys, why these human beings have built up this transportation system, which is, the very basic question, why is that being built? Then you might come to the conclusion they built this to transport one thing to another place. So to move persons or goods from point A to point B, right? And so then if you, with this answer in mind, if you think about the materials that maybe were at the very beginning of transportation systems, so for example, when the Roman Empire started to build up roads, maybe it won't be that difficult for you to reconstruct or to think about what a road at that time could be. And this is actually a picture of my mom's hometown. This is Pedre, nearby Pontevedra, nearby Santiago here. And this is Roman Road. And you can see that at the end of the day, it was just flat blocks that were connecting Pedre with Rome. So to move some goods or some soldiers or whatever they wanted to Rome. And so that's why we try to reconstruct these functional systems with simple building blocks. Sorry. And obviously, we have to focus on a particular function. And for us, it was very interesting to see the discoveries at the late 90s of filaments in archaea. So there was these papers appearing in where scientists discovered that the oldest cell in our planet, they have a cytoskeleton, right? That was interesting. And these first discoveries were followed up many more other discoveries of different scientists founding this kind of one-dimensional supramolecular polymers in many different old cells present on the Earth. And now it's accepted that most, if not all, bacterial and archaea cells contain at least one protein filament system. That was kind of not well accepted because people were thinking that maybe the cytoskeleton was some sort of a late in evolution organelle. But if those kind of supramolecular polymers have been there since the beginning, that means that they could have been an advantage for the early protocells. And therefore, this advantage was kept over evolution. So the basic question or the basic idea that we have at the beginning was, let's try to replicate the supramolecular polymerization in confined systems. So when we went into the literature, obviously there has been a lot of previous work, but mostly using natural proteins. So here you can see a giant unilaminar vesicle which has actin reconstituted inside. So basically you have all the cellular machinery, you add the ingredients, and the actin fibers are synthesized and are self-assembled, and they get this shape in an artificial compartment. Then you have some sophisticated systems like this. This is a water in oil droplet which has microtubules which have kinesins that are connected and walking over these microtubules. And you can see that when the system is saturated, the microtubules assembled into a nematic phase at the cortex of this droplet, and they move as a perfect tetrahedron on the cortex of these water in oil droplets. So despite those many examples in the literature using natural proteins, natural monomers, there was not so much using synthetic systems such as simple molecules as the molecules I will show you in a minute. And so we started this project or this research line with this basic idea: try to replicate that supramolecular polymerization phenomenon in artificial systems to see if they could lead to any kind of functional response. Here there are some of the natural roles or natural functions that the cytoskeleton is playing. This is an amazing organelle, very complicated. There are several examples here, the metaphase, here the example I showed you before. This is a very nice example of asymmetric distribution of fibers in giant vesicles. And this is another very interesting example of how nature can regulate the formation of one-dimensional supramolecular polymers. These are examples I just found in the literature over the years. This is, I think, actin fiber reconstituting at the nuclei of the cell to help in DNA damage repair. This is really amazing because it means that the cell can, move across all the membranes and all the cell, the actin monomers, so that they are only polymerized at a certain moment in time there in the nuclei, which is really, really surprising, the level of the control, the degree of control that the cell can achieve for these processes. And to start with a simple molecule, we decided to use these building blocks here, which are really very versatile, they are called self-assembled cyclic peptide nanotubes, as introduced by Professor Reza Gadiri at the Scripps and Professor Juan Granja, my colleague here at the Ficus. And they found that if you have a cyclic peptide made of an even number of amino acids of alternating chirality, such as DL, 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 then this particular configuration will force the ring to be planar and stacking on top of one another by this hydrogen bonded network. And so creating this type of nanotubes, which will have, in which you will have a very nice degree of diameter control, because you can simply add or subtract a number of amino acids, and also the chemical decoration of the exterior, because as you can see here with this alanine residue, if you put the side chains of the amino acids are radiated outwards of the tube, so you can change both thickness and external chemical decoration. We've been playing around with these systems for a while already, preparing hybrids with peptides and carbon nanotubes. Here we wanted to use that as just a model of a one-dimensional supramolecular polymer in an artificial compartment. So for that, we designed this peptide here, which is again using the hydrophobic effect as the main driving force for its assembly, but it's an amphiphile. It has a hydrophilic part here with histidines and the lysine unit, and these histidines can be protonated or deprotonated near physiological pH, so that you can control the self-assembly. So if you have that under acidic condition, will be deprotonated. If you add the base, then they will deprotonate the histidines, the peptides will come together and will assemble into this kind of structures in which they will align the pyrene units, which are the hydrophobic moiety of the molecule that we have made. However, there's here a mismatch, because this is an antiparallel Vera sheet, which has a distance of 4.5 angstroms, and here we have a pi-pi interaction, which is a bit slower, a bit smaller. And so this mismatch was ideal to get interdigitation of the pyrene units, so that we can get fibrillar bundling. And this was one of the first ideas that we go from the nanosystems to micron scale fibers that we could observe into our microscopes, that we can, check the formation of fibers with the right size and kinetics as the cytoskeleton in living cells. The first thing we did was to put this in water at sufficient concentration. When you add base, for example, sodium hydroxide, deprotonating the histidines, assembly took place, entanglement was there, and therefore the water solution became hydrogen. And we could check also the fluorescence intensity of the pyrene unit, which is confirming the assembly. Because when the pyrene it's assembling with other pyrene units, its fluorescence emission changes. So the band is quenched, the band of emission, the normal band of the monomer emission is quenched, and a second band of an hexamer emerges, right? So basically it goes from the blue to the red, or in this case to the green, so to say, right? Because it's not going really far to the red, but it's going from the blue to the green, so we can check that type of self-assembly by looking at the pyrene fluorescence. Here is circular dichroism. This is a measurement of the chirality of the supramolecular structure. You can interestingly confirm that the pyrene was sensing the chirality, so it was assembling into a chiral environment, and you can plot this in front of the pH. As you basify the system, it gets assembled and it's getting a more chiral configuration. And if we plot this intensity against the pH, we can check that we have some sort of a cooperative supramolecular polymerization mechanism, which is nearby or closely located at the pKa of the histidine unit. Microscopy for the nanoassemblies, you can see here that we have atomic force microscopy with tubes of around 2.5 nanometers, 3 nanometers, and also in the electron microscopy you can confirm the nanostructure. But we wanted also to see how this nanoassembly was propagating, and if we would have entanglement over mesoscales and in the microscale. And well, first we've measured rheology. We could confirm that we have hydrogel with viscoelastic properties, which was not so much surprising in the standard range. But the interesting thing was when we took part of these layers of the gel and we observed them under the microscope, then we confirmed that we have the fibers that we were looking for. This is imaged by using thioflavin, which is a probe, a fluorescent probe, that is planarizable, so it rotates. So once it gets planar and it's for example, in the hydrophobic effect, sorry, in the hydrophobic pocket of a supramolecular polymer, such as a beta amyloid or our polymers, it will shine, it will emit light. So you get rid of all the background fluorescence, and you will only get the light signal when the fibers are formed. And that was very interesting for us. We could confirm that we have nano assemblies that were bundling into the micron scale. And so then basically the next thing was just to put this in droplets. And so we put these monomers here in a water in oil suspension. This is water, this external layer is oil, and we are a base. Just make an emulsion and we are a base. I think it was a small hydrophobic amine that could navigate the oil and check and enter into the water droplets and see what happens when we have them there. This is epifluorescence image. Here you can see the monomer under acidic condition. You can see the emission is blue, so kind of well-solvated monomer solution, and this is confocal projection. There is nothing really going on. But when we at the base, already at the beginning, we could observe the formation of these fibers, micron size, which already, as you can see, they have this nice green shift kind of emission, right? As I mentioned before, the pyrene is getting stacked, and you can see the excimer emission there. And this is confocal projections. You can see fibers are there as well. Here it's, we didn't have the chance to make videos at a time because our microscope did not allow, but here is the addition of propylamine here, and then you can see how the fibers being formed, and close to the addition point of the base, you can see all the fluorescence, all the green shifted excimers being formed, and far from there, still plenty of monomer being dissolved. And well, we also characterized this system, and we found out that whenever there was a fibrillation happening, there was a change in the spherical shape of the containers that we were using. And well, this is a kind of a representation of the Maeda actin fibers with our droplets, which are not that different. This is a very old confocal, not easy to see, but this is confocal reconstruction made by Alejandro Mendez on one of these droplets. You can see that the fibers are distributed all over the droplets, right? And so next, what we wanted to do is to try to control a little bit the positioning of these droplets. And for that, we used one of the most basic lessons in supramolecular chemistry, which was to change the trigger that was causing the supramolecular aggregation. So as I mentioned, we use basically at the beginning, we have these monomers in acid. We could add a base so that this will deprotonate and come together because the cationic repulsions will be removed. But another way to get this assembled was to use salt. So if you increase the ionic strength of the system, then also the counter ions could shield the cationic repulsions, and you could get the fibers together. If you again see that from this top view, you will see this kind of system. And what we were thinking was that if we could, trigger the self-assembly by using base, deprotonating the tubes, we will have tubes which will be hydrophobic. So maybe those tubes could change. I mean, and if we use salt, the tubes that we will be assembling will be more hydrophilic, maybe more prone to get assembled into the core, where there is more in the aqueous compartment of the droplet. And if we trigger the assembly by base, maybe those will be more at the interface where there's the oil phase, which is more hydrophobic. First of all, we checked if we could trigger the assembly by changing these two conditions, and we found out that yes. So here is the pyrene emission, and if we use either base or either changing the salt, we could quench the emission of the monomer and get some emission of the excimer. Excuse me. So what we found was that the base was triggering a stronger quenching and higher excimer emission than the base. And this is kind of expected because if you trigger assembly by deprotonating the histidines, then the supramolecular tubes will be will be well packed, less protonated, and easily packed and more closely packed the fibers, so the pyrenes will be closer to each other. And again, we could confirm that by UV as well. So here, what we wanted to do is to have a very good control of the trigger of the supramolecular polymerization of our monomers. And for that, we implemented microfluidics. We did most of the work here, but some technical work also we did with our great colleague, the late Professor Chris Abel, who I would like to remember. And so we have the very simple microfluidic system, which is the peptide will come here in aqueous solution. The oil will come from here, and we will generate droplets, uniform droplets. And then we could, repeat the same as we were doing here with our simple microfluidic systems. And with Chris, we have more sophisticated systems in which we will have already the peptide droplets coming from here. This is just the peptide droplet, the aqueous droplet, with acid coming from here, and then we add here this base pico injection. These are electrodes that facilitate the fusion, and we could control the addition of base with high precision at this particular point. And with this, we could, play around a little bit more on how we were gain control on the trigger of the supramolecular polymerization. And well, that was it. We wanted to have dispersing peptides, and we wanted to check if by controlling the assembly triggers, we could move those two situations. So here is the system. We have the oil here. We have the two waters. We have the water here coming, and then we have here the trigger for the stimuli to trigger assembly. Now you can see that the droplets that we make are all equal. We could prepare a lot of droplets, all of them the same size. And here is the efflorescence, and here is the profile, and this is confocal microscopy. You can see that the monomer is around and is well dispersed. When we use base, when we trigger the fibers formation with sodium hydroxide, we could check fibers were at the cortex. And when we use calcium chloride, we could observe that the fibers, as we were expecting, that the ionic shock was triggering these hydrophilic fibers that were assembled in the core of the droplet. Sorry, the confocal at that time was not very nice. So we have, but was clear that they were there, also in the profile. One idea from Alejandro, which was amazing, was maybe if we could control by the ionic strength the core assembly, maybe if we could control the flow rate of the salt, maybe we could even control the positioning of the droplets. And this is what we did here by just controlling the flow rate of sodium sulfate. We could control the how close to the core or the cortex the fibers are assembling. So if you do a strong injection of salt, then everything goes to the nuclei. If you start to decrease, everything goes to the or it's more heterogeneously, more homogeneously distributed. Once we were there, we wanted to see if we could, do something with these droplets. When we could control the assembly at the core or cortex, maybe we could do something to control the functionality of these droplets. And for that, we just check assembly of the droplets, how the droplets get together in different situations. So whether they are monomeric, whether the fibers are at the cortex, or whether the fibers are at the core, right? This is monomeric. When there is the monomer there, the assembly of droplets is a bit chaotic. You get some coalesces, some aggregation, but nothing really other. When the fibers are at the cortex, what we observe with this huge green shifted, this is a really massive aggregation. So they are already there, and when the droplets get together, this is massive aggregation of the droplets in this kind of proto tissues or systems like that. But when we did the assembly using the fibers, the droplets that have fibers at the core that were slowly diffusing to the cortex, we got this kind of almost perfect hexagonal networks of droplets getting together. And this is a funny picture when we have the droplets at the cortex, but they couldn't get together and they were dry. Basically, we could find that the fibers were expanding into the direction of how the droplet was collapsing in the plate. This is kind of a funny picture, but wouldn't you tell me that some of those resemble like a actin fibers network covering the nuclei and extending to the exterior? Well, anyways, One of the before, so all this time we were using a monomer under confinement and changing the external conditions, but we wanted to do something to trigger the formation of the cytoskeleton of the so say protofilament systems by using a chemical reaction. Because this is amazing video from the Nogales lab, and there is a lot of amazing work on this highly complex and really intriguing and sophisticated process from the assembly disassembly of the cytoskeleton of cells. But most of these or all these very complex phenomena happen just by a phosphorylation or a dephosphorylation reaction of a protein monomer. So obviously we are not biochemists, we don't work in this field, but we just wanted to replicate with a very simple reaction the formation and the self-assembly of a protofilament in a confined environment. For that, we changed the building block, and for this we used these peptide amphiphiles that were introduced by Professor Samuel Stupp and very beautifully reviewed here in these accounts of chemical research. And what they did was they discovered that if you have a peptide ending up in a hydrophobic tail, so here this end could be, I don't know, 8, 10, 12. It's a long aliphatic tail, right? So they discovered that these kind of peptide amphiphiles were capable of self-assemble into solid fibers, and that they were bonding together into micro assemblies as well. Sorry. So here, the structure of this molecule is very simple. So that was a kind of advantage for us to start working in this field. You see here, this is a peptide which has two anionic residues. In this case, it's glutamic acids. They are anionic, and this is important to remember. Then there is a region which is considered to be the beta-sheathing region. So these amino acids here, glycine, alanine, and valine, will help the peptide to self-assemble into beta sheets, which will trigger the formation of the nanotubes. And then this is the aliphatic tail. And what we brought here to just screen for the right structures was that we were making the connection between the peptide and the tail using an oxime bond formation. So basically, this is you start with alkoxyamine, which is just this here without the tail, and you add an aldehyde. This bond will be formed. This is an oxime bond, and in this way we could, check for many different combinations of peptides and heads, of peptide heads and tails, so we can maybe find out easily what were the best structures for us to start trying to replicate this system that could self-assemble into a nanotube and then into a microfiber with the right size and kinetics for us to image them into the microscope. And well, we tried several combinations of heads with different number of amino acids and different aliphatic tails of different length. And to make a long story short, we found that while the system was kind of well tolerating at the peptide head, the tail has a preference in this case for tetradecanal. So basically here we were combining a peptide head, like this one here, with a tetradecanal tail. So the tail, the highly fatty tail, was 14 carbon atoms, right? So we did several experiments and we found this particular combination which was good, and we could find, there were some others, but this one was nice. For example, here you can see nanotubes with a P5T12 as well, nanotubes that then were assembled into micron fibers. Again, imaging them with thioflavin, right? So after that was discovered, we decided to make this connection between the peptide head and the tail in water. So here we were just making the peptides in the MSO and checking where were the ones that were good for assembly. But now we wanted to prove this thing of connecting the two things separated in water. It was not easy at the beginning, basically because the tetradecanal tail, so a tetradecanal aldehyde, was not soluble in water. It took us a while. I always say it, with some smile on my face, because it took us a while to notice that a simple solution for that was reducing the number of atoms of carbon atoms of the tail. So we did that by incorporating part of the tail into the peptide head. So in this case, now we have the peptide head with some with part of the aliphatic tail already incorporated in it, and then we combine that with the aldehyde. In this case, it's octanol, which was more soluble in water than the tetradecanal, of course. First experiment we did was measuring fluorimetry. So we mix these two components in water in the presence of thioflavin. This probe I mentioned that only emits light when it's in a hydrophobic environment and it gets planar. And you can see how after the mixing of the two compounds, there is an increase in the fluorescence, which was already a good indication that the two components were reacting and assembling together. So then we measure circular dichroism. Again, as I mentioned, when the system gets assembled into a chiral environment, you will get a CD signal. And here we got it, and we started to measure it and to plot it against time. And here we could observe that the dependence of this CD signal was acquiring this kind of sigmoidal shape, which was, as described by many before, clear indicative of a physically autocatalytic process. And so what we were thinking was that these two components, after reaction, they will form in these aggregates, which we confirm by different measurements. And after the formation of the bigger amphiphiles, so basically after the increase of the hydrophobic character of this peptide, the amphiphile was self-assembling into this aggregate that were increasing the solubility of the two components and bringing them into close proximity, accelerating their connection. Then, after that, we will get the nanofibers being formed, and then they will be self-assembling together into these microfibers, as mentioned before. To confirm the physical autocatalytic process, we check HPLC for the disappearance of the peptide head. So basically, we monitor the reaction, we monitor the amount of the peptide head by HPLC, and the typical process like this follows initial lag time. Then it's then kick off the project, the reaction, and then you can see the reduction in the amount of the peptide head. And this lag time that you have in here could be reduced or eliminated by adding 5 mole% of the final product, which is acting as a catalyst of its own synthesis. And so that confirmed the process that we were suspecting. And when we look at the bulk in the water, we found these fibers, these aster-like fibers from these nucleation points, which were very promising and highly encouraging us to follow up on the encapsulation of this monomer here and the external addition of the aldehyde to check if the system could be fibrillating in water droplets. So again, we make an emulsion and we encapsulate the peptide head, and then we add the aldehyde from the exterior, and we started to check characterize the system. Here again is fluorimetry, and you can see that the system follows this sigmoidal shape with this lag time that we can reduce or even eliminate by adding increasing amounts of the seed of the final product of the reaction. We did some controls to confirm that the amphiphilicity was fully required to get this self-assembly process, and that the hydrophobic the character of the molecule has to be fine-tuned for this process to happen. In the droplets, when we add the aldehyde here, these are droplets being formed, and you can see after addition of the aldehyde here in the oil phase, which is diffusing and reaching the droplets, you can see that after some time, the abrupt emergence of these fibers, which as shown here, they appear to be formed homogeneously all over the place, but with the time, they start to nucleate and accumulate at the cortex, which was in our case rationalized by the fact that the cortex of the droplet is the space where they have more space to grow longer, and at the same time where more aldehyde will be concentrated so that they can keep consuming the aldehyde that they need to keep on growing. This is a video here, maybe you can focus. We bought a new microscope so we could get better videos. So here is the bundling and the uptake of a smaller droplet. Here you can see these events of coalescence. Here you can see the fibers, how they get bundled together, and how some droplets were like taking up the other ones, the smaller ones. And you can even sometimes see how the contents of one droplet was like being sucked by the other who was nearby. So this kind of thing, of seeing fibers interacting at the cortex of droplets and how do they were interacting with the exterior, trigger us the curiosity of checking if that kind of communication could enable cross-coupling or cross-communication between droplet populations. For that, we use the typical assay, which is glucose oxidase and whole-reduced peroxidase. It's an enzymatic cascade. In this case, what we did was to encapsulate the enzymes under products orthogonally. So here is glucose oxidase, and here is glucose, and here is whole-reduced peroxidase, and here is amplest red, which is a molecule that will be oxidized by the hydrogen peroxide produced by this enzyme, and after oxidation, it will produce fluorescence. And basically, we put this thing, we label the glucose, we use two droplet populations, and we label the glucose oxidase in blue to check what kind of communication did we have. And then we add the aldehyde. And so we have this maybe contact, bundling, exchange of substances, and if the glucose will come here, will be oxidase, will be the whole cascade could proceed, and at the end, the final readout will be the oxidation of amplest red in the droplets that will produce red light. Here is the video, and you can see that after we add the aldehyde, emergence of the fibers, and in the droplets which were more, so to say, getting red color, you can see also the higher amount of green fibers labeled with thioflavin. If you see that the droplets were turning red before the fibers could appear, it's explained, this phenomenon is explained because we could only observe green fibers when they are becoming big, when they are micron in size. However, we should keep in mind that before that, they are already nanometer fibers being formed all over the place, and that were enough to trigger the communication between the droplets. So that's why you observe the red dye, the red color before the green. Oops, sorry, I will play again the video. Here, as you see, this is one of the one that starts red faster, and you will see that after that is going to be one of the ones that has higher amount of fibers and is even taking up other droplets in the surroundings. Interestingly, we found that although we have exchange of the small molecules, we did not have exchange of the big protein. So the blue droplets remain blue. They did not exchange the blue compartment. And all these things trigger us to check if this process could be related to the uptake of exogenous compounds in the similar droplet. And for that, we did experiments of droplet of supramolecular fibrillation in the presence of how am I doing with time, Evan?
5 minutes left.
All right. So I'm okay. Okay, so here we trigger the fibrillation in the presence of different probes. Basically, we took some of the probes that we have around in the lab, and we did the fibrillation in the presence of these molecules. And remember, our fibers were anionic. And we tested fibrillation in the presence of a sweeter ionic, rhodamine B, a cationic dye, rhodamine 6G, and another highly cationic dye, OH, which we used to also to label DNA in living cells. So here, in the first one, you can see that after adding the octanol, fibrillation was happening, and the fibers were taking up the amphiphilic compound. But beyond that, nothing was really happening. However, when we put the cationic dye, the uptake was also present, but at the same time, it was accompanied by a strong degree of coalescence of the droplet population. And that's easily understandable because you have a cationic dye which is getting in between the anionic fibers, minimizing the anionic repulsion, enhancing bundling, minimizing surface tension, and maximizing the droplet coalescence. And we found that this also with OH, as you can see here, OH is in blue and the fibers are in green. And you can see how the fibers start to coalesce and even rotate sometimes to maximize the fiber-fiber contact, as shown in this couple of droplets here. Well, anyways, take home message. The simple message here is that a slight increase in the hydrophobic effect by making a simple reaction on a peptide amphiphile could trigger very complex uncoupled phenomena, like a simple oxygen bond formation can trigger the fibrillation, the uptake, the selective uptake of different things, the bundling at the cortex, which then can in turn control the coalescence of droplet populations. And this project has not stopped here. We continue that thing with the help of many, some of them already in the audience here today. We are moving from water in oil suspensions, which are a system that we still like and that we can continue to investigate. But we are trying now to move to water in water systems. And in this case, it's dextrin in PEG. So we have condensates of dextrins in water, so water in water droplets. And there is no confinement in a water in oil kind of border, kind of boundary. And in this case, to increase the solubility of every component, instead of using octanol, which is the aldehyde that we were using before, we use hexanol, which is an alcohol, which is way more soluble in water. And we use an aldehyde to convert this alcohol into an aldehyde. And so that we can control the fibrillation by the enzyme kinetics, right? So the enzyme will convert the hexanol in hexanol, the peptide will react and will form the fibers. And you can see here the video. It's again, fibers are there, but the behavior is a little bit different. You can see some funny things happening all over the place, especially some, I don't know, fibrils being formed. But normally, we always, when we are focusing our videos, we just focus on the things that we see and not on the things that we don't see. So when we could, stop the video and take a picture of these things, we can see that the fibers are also now projecting outside of the droplets and of the condensates. And we are now carefully studying why this is happening, and if we could control that by or even use it, or what are the implications of this kind of supramolecular phenomenon. And very soon this will be out, and many more things will be developed. This is the last slide a scientific slide that I wanted to show you on our efforts to try to understand or to get some feelings on what a amphipathic fibrillar system could have done at the very early stage, or how could we even implement these kind of things in functional synthetic cells. And this is a very old picture. This is not all my group, of course. This is a group excursion to some island. And here there's plenty of people that are still on my lab, many of them that are not belonging to my lab. These kids are not belonging, and many friends who were there. But this is the picture in where I found most of the people involved in this work were around. This is Richard Both here, Alejandro Mendez here. He's one of the most important contributors to this research in my group. He's now a professor in Sevilla, in the University of Sevilla, in Organic Chemistry Department. Nacho Insua here with a short haircut, and Richard Both, who's now in the Penn State in the States, and Sanawad, who's fighting to get a position in Calcutta. And we are here at the Santiago de Compostela. This is the Santiago's Pilgrim way, from where everybody walk from many distances. You are all welcome to pass by anytime. I want to thank, of course, the funding agencies for this work, especially Human Frontier Science Program, who funded a very crazy idea that it's still being proved or still it's some more time to be demonstrated. Maybe with the help of the coming cell, we will reach there sometime. I also will never have to forget the ERC, the European Research Council, which is really transforming the science all across Europe. And of course, I would like to thank you, Evan, for organizing all of these series of lectures, and all of the virtual attendees for your kind attention. Thank you very much.